So Dr. Manisha, we will be recording this meeting and we will be sharing it. So those of you who are interested in listening to it later, we will be sharing it. Okay, so good evening, everybody. This is Rajvi here, and I would like to welcome all of you to the Journal Club of the Academy of Clinical Embryologists. We have got a very good response to this Journal Club, one of our academic activities of ACE. So friends, we all know that plastics and nanoplastics, microplastics have created a havoc in the environment. There is possibly no body fluid which is free from these plastics. They have been detected in breast milk, urine. So there is a big concern about plastics. And as embryologists, we are all extremely concerned, about the quality of plastics used. So when we started, we would only take care of endotoxin toxicity, endotoxin tested, then we would have plastics which have been tested for tissue culture grade. Now we have our disposables which have been checked for embryo quality by mice embryo testing. Now the question is that are these plastics safe? And we came across two very interesting papers. Both have come from France and with collaborative, one of them with collaboration with Tunisia, about the impact of plastic disposables that we used in ART. And for this discussion, and they have been in high-ranked journals, so it's not some off-shot paper. This paper has been published in Human Reproduction. So do plastics affect embryonic development, and do they affect gene expression? The good part is the gene expression paper has not been done in humans, but it is a laboratory model. And I'm directly going to switch over to Dr. Charulata Chaterjee, who is going to give us a brief of both these papers. And subsequent to that, we'll have Dr. Nishad and Dr. Sarguna Devi, who will initiate the discussion. And all of you are welcome to put in your questions in the chat. And at the end, we may even take oral questions. So without much ado, I hand over the proceedings to Dr. Charu Chatterjee.
Thank you so much, Rajvi, for giving us, and I would like to thank ACE for giving me opportunity to discuss such a interesting subject. I myself has done lots of, want to do lots of reading for this. And shall I share my screen now, Rajvi?
Yeah, please go ahead. It's over to you.
So we are discussing the two very interesting risk papers. Both, as Rajvi told, it is from the group of France, that potential effect of disposable and plastics in IVF. So as an embryologist, its slide is not moving. As an embryologist or ART specialist, for the goal for all of us is to minimize the risk of multiple birth, and for that now a single embryo transfer, preferably blastocyst, what we are preferring. We have to optimize pregnancy rate and produce healthy, genetically normal, a full-term delivery. So here two critical questions arise in the mind, that are we doing harm when treating patients, gamete or embryos in ART? And do the ART treatment per se cause any adverse outcomes? So these are the questions comes in everyone's mind. And this is the very interesting subject which has been published recently in a Human Reproduction 2024, that disposable use cumulatively in routine IVF procedure could display toxicity. So here in this study, what they have done is very 14 categories of consumables what we routinely use in the IVF laboratory. They have, and this all the disposable, when they come into the laboratory, it is tested for the toxicity test. And when single they have tested, it was non-toxic. But in the combination of 2, 3, 4 or 5, how they behave, do they toxic when they are means we are using in the combination? So that different combination is used here for this test. And the test what they have done is human sperm motility test and the mouse embryo test, and they have done the SMI, means sperm motility index, which they have calculated statistically 0.85 and above will be the toxic. So first and premost things, like when we are talking about the plastic, let's start from the plastic, what we use into day-to-day life. And we all know that there are lots of things which is done made up by plastic and we use day to day. So what is plastic? Plastic is a kind of, it can be from the natural source or it can be from the synthetic source. So synthetic source is made up of polymers. So polymer actually is a Greek word, means many, and the part. So large molecule or macromolecules composed of monomers. That is the definition of the plastic. And the plastic, there are three types of origins: the natural polymers, as I told, synthetic, and the semi-synthetic. Based on the classification done by structure and functional group, if you just little have the idea, that linear polymers. So linear polymers are the example of polyvinyl chloride, what we know. So monomers here join in each other to form a long straight chains. Whereas there is another plastic which is also called low density polyethylene, that is branch chain polymers. And the other, what routinely we use in the lab, polystyrene and all that, which the dishes are made up of, it is basically a cross link or network polymers. So these polymers are formed by bifunctional and trifunctional monomers, and that is what we are using into our laboratory. As per the chemical process, additional polymer, condensation polymer, and cross-link polymers. So if we give the classification by its physical property, then because it is easily moldable, it can be, the density, hardness can be maintained, and then it means property of plastic can be made. So this is a plastic. So plastic is nothing but a chain of identical molecules called monomers. It can be high density polyethylene, low density polyethylene, polypropylene, polystyrene, and another is PVC. So these are all a different type of plastic. So we are using is polypropylene and polystyrene mainly. So these are all the polymer and its uses in our life. So what we are using in the lab is what we are discussing today. First, let me see about the dark side of the plastic. So this plastic is releasing the toxic chemical into the environment. Sometimes in our IVF lab, it is responsible for hormone imbalance, and long prenatal exposure can also affect the offspring. So combined effect, like, VOC, we all know that accumulate into the incubator, it can also alter the culture conditions. And that is the reason the ASRM and ISHRA has given the guidelines that what kind of plastics we are using and what quality check every embryology should check before taking that disposable or plasticware into the lab. So the aim of this procedure, this study was done in France for almost 1.5 years. They have used the surplus semen sample with informed consent, and the sample is, as per the WHO, it should be normozoospermia, and they have prepared the sperm by double density gradient, and the post preparation should be at least 10 million with 70% of motility. The final concentration of the sperm they have adjusted to 4 million. Here they have included 14 categories of disposables in this study. So what kind of disposable used for these studies is cups, condoms, oocyte, condoms means without spermicide, oocyte aspiration needle, manipulation flask, tubes, tips, pipettes, embryo transfer catheters, syringe, and gloves. So, and even the straws. So what they have checked the toxicity test by human sperm motility assay. So all we know that we were also doing the sperm motility test to check our kind of new lab and function of the incubator or function of our disposable. So we all are aware about the human sperm motility assay. So initially, when I have started this sperm motility test, we were doing for 96 hours, and then we were checking the survival of the sperm. But for this test, the sperm SMI or sperm motility index, they have for 24 hours at room temperature, and 50% motility was the cutoff for the test what they have used, and 0.85 is the absence of toxicity. So these are the picture says that how they have checked the different combinations of disposables for this study. So picture A says that from the sperm, prepared sperm from the tube, one will go to the control tube, one will go to the 30 minutes exposure into bottom of the petri dish, then again 30 minutes exposure to the lead of the petri dish, and then it will go to the test tube. So this is the one combination. In case of two or three combination, they have used the pasture pipette or the transfer pipette, then the bottom of the test tube, then the lead of the test tube, subsequently bottom of the petri plate, and lead of the petri plate. Another 4-5 combination, petri plate, tubes, syringes, and pipettes they have used for the combination. So these are the things how they have done these studies. Here this are the whatever it is coming into this study, individually they have all passed the toxicity test, and control is always non-toxic for the anything. So now this whatever material has come in, they have checked except few. So it is MEA certified, human sperm motility assay certified, and CE marking. CE marking is for the European country. They have the taste for the European conformity. So that also they have checked here. So these are the various 14 categories of disposables. So statistical analysis they have done by the CMH and another parameter. So that, and they have indexed that SMI is more than 0.85 will be non-compliant for this studies. The sperm motility index is so 36 references and 53 lots. So the 49, that is 92% of item, and they have tested this taste in the triplicates, not in a single twice but triplicates, so that is non-toxic. But 4, that is 8% exhibit toxicity. So out of these 4, 2 were marked without MEA and HSMA, so that product can be ruled out. So that is, and the modified alternative this extraction test, they have check the lead when they have some kind of FDA approval. If you check, so they test the only test tube, they don't check the lead for the toxicity test. So when you check, do the test by keeping the sperm into the lead of the test tube or a cap of the test tube or the lid of the petri plate, it will show the toxic. And surgical gloves they have done for 1 minute, 5 minutes, 10 minutes, and 30 minutes. But if you keep the sample more than 10 minutes into the surgical gloves, it has exhibited that it is a toxic. So when this have done for the combinations of consumables, they have used 48 combinations, 41 lot of 32 references, which has passed individual test. So out of this 48, 17 exhibit SMI below 0.8 for 2 of 3 tests. And out of this 17, 18% 3 consumable association, where we have used 3 category of disposable, and 5 out of 16 means 31% is 4 consumable association, and 9 out of 15, which is really worrisome, which is 60%, where we have used 5 more than 5 or more than that consumable for the procedure from pickup to transfer. So definitely we all are using more than 5 kind of various disposables into that. So in that frequency, 35 mmds, XCDs, 100 mmds, and tubes, and pasture pipette. So tubes showing 29 and pasture pipette is 14.9%. So this is again, this I have done it. So the conformity with 3 consumables, you can see compliant, but conformity when a 4th consumable was added, you can see that non-compliant is, and but when you see the conformity when a 5 consumable was added to the previous association, then the toxicity is more. So coming to the discussion part of this paper, the precise source for toxicity, maybe it is unknown, but maybe we can assume that various sources like material composition, production process, handling and packaging, sterilization or transport process. So anything can go wrong and the product will show the toxicity. This material reduce or stop cell metabolism, and it also responsible for motility. So motility also decreases with the time increase. So for embryo, we may observe the reduce or decrease fertilization rate, blastocyst rate, and it affect negatively on post implantation and health of the newborn also. So the susceptibility of toxic may differ among biological samples, but these days all the supplier or manufacturer are aware of the safety, reliability, and the traceability. So from gloves, we can all feel that why gloves is in a 10 minutes is toxic, because gloves has a fine particles which may allow toxicity to come in. As I also earlier told that some kind of FDA guidelines will, taste the only the part where the biological material comes into the contact. Like petri dish means only bottom of the petri dish. Test tube means only bottom of the test tube. So they don't check the lid. So when we have checked for the lid or a cap, that has showed the toxicity. So the IVF material made up of polymer from thermoplastic family like polystyrene, polypropylene, and polyethylene. But again, what we have discussed earlier, that material made up of polycarbonate can leach BPA, which is very, very toxic, like bisphenol. So we all are aware that how toxic bisphenol, not only on developing embryo, but also on the male and female reproductive system. For male, in the reproductive system, it can reduce the leading cell function. It can also affect the pituitary, hypopituitary gland. It can affect the sperm production, the motility, concentration, and everything. And so in the woman also, it can also affect the uterine morphology. It can disrupt the menstrual cycle, and it can also disrupt the hypothalamic pituitary axis. So this is the kind of effect of bisphenol on the reproductive system. Coming to the embryo, it also has the very negative effect on the embryo. So limitations for this study, what is the limitations? Because here they've used the limited salmon samples. So when they have tested for the duplicates or triplicates, though the same sample may not be available that time, but though they have checked the homogeneity of the sample. So again, this test continue for many days, though into the controlled IVF laboratory, but it cannot assure the any minor or major change into the temperature and humidity. So that cannot be ignored. So operator variability in spite of internal and external QC check, because the tests run for so many days, so there are different operators. Though the operator efficiency they have checked, but that cannot be, the error can happen in the operation also. And mechanical error can happen during transferring of the semen samples, because here we are handling a minimum volume of the semen sample while transferring. Though we are use a device which is QC checked and all that, sometimes there will be a leftover semen sample into the previous tube or previous transferring tube, that cannot be also taken into the consideration. So these are the limitations for this study. So conclusion, coming to what I understand from this paper, as every embryologist should check the toxicity check before use or before taking any new disposable into the laboratory. Human sperm motility assay should be regularized and standardized, rationing the number of consumable used in the each procedure. So that how every embryologist should learn that how we can use the minimum. Now this paper also give me a kind of thought in my mind, that how do they dispose all these plastics and all that, when it is such a hazard not only for developing embryo, but after that we are using bulk and bulk tubes and dishes and all that. So either they can be recycled, they can be burned, or they can be use a plasma pyrolysis technology to where the organic compound disintegrate into gas and into oxygen starved environment. So this is one of the new technology, and I think the plastic engineer should have the better can do some work for us and shape the future of plastic industry, not only serve the surroundings and our aquatic animals and our day-to-day life, but for the developing embryos into the IVF lab, there should be some kind of blend of the plastic that can be have a minimum or a no toxicity into the lab. So the plastic, we maybe depend on the plastic engineers into the future. So this is the discussion of my first paper. Shall we take Rajvi question at the end of the each paper or end of the two papers?
I think we can do it because both are interlinked in
Yeah.
a way.
Okay.
So,
So yeah. So then the another
and
interest. Yeah.
maybe the audience, you can put in your queries and questions and thoughts. So more than questions, it will lead to a discussion. So please
Yes.
feel free to put your thoughts down. Over to you.
So again, there is a very interesting paper which is also recently published in April 2023, and a group of France, that plastic used in vitro fertilization procedures induce massive placental gene expression alterations. So these studies they have done on the animal model. So they have used a hybrid mice model for the study of placental gene as well as brain gene, and carried out a different kind of assays how it affect the gene and that. So now if you study that in the regular conception, about 3% of live born will have some kind of major to minor anomaly. And why it happens, that we all know, maybe because of the genetic or maybe because of the environmental factors. But if you calculate means talking about 3%, it is very less. But if you see the 3% globally newborn, the count is so huge that this many baby is born even after the natural conception with some kind of anomalies. So burden is not only on the birth defect, but there are kind of emotional, social, financial burden. uh, Patients should go for elective terminations, stillbirth, survival and comorbidity, long-term disability, quality of life, and the economic cost involved in it. So these are the kind of incidence and prevalence for the natural birth where the child is or a newborn is having some kind of anomalies. So we all know there is maybe genetic cause, single gene defect, chromosomal defect, numerical or structural. Environmental will contribute 4 to 10%, multifactorial with 20%, but there are unknown which contribute about 60%. So but in case of art, where patient is going for ovarian stimulation with using of our plasticware and culture media and another manipulation of the gametes and embryos into the lab, might also lead to kind of disturbance into the developing embryo. So the in 45 years of IVF now we are having. So the first risk of IVF baby, which is documented, that the low body weight, and the baby born through IVF is 2.6 times having more incidence of having a low body weight as per this paper. And this studies has done for kind of 2 crore 53 lakh 5,056 women, where they have checked for this maternal risk, which has included morbidity, mortality, hemorrhages, severe preeclampsia, and acute renal failure. So for this reason, they have kind of done this study on the animal model, and 6% of transcripts altered more than twofold in placenta. So this study has done for the transcriptome alterations after incubating embryos in plastic as well as glassware as well as in vivo. So they have taken B6 by CBAF hybrid female. The female mice is 5 to 8 weeks old, whereas male mice is 30 weeks old. Here, group A1 and 2 is IVF embryos. All the embryos has developed till blastocyst, and at the same stage blastocyst, they have taken the blastocyst, expanded blastocyst, not in the morula or a slow growing blastocyst. And all the embryo culture medium is same for plasticware as well as for the glassware. So ovulation induction protocol remains same. They have used 8 IU of postmenopausal semen serum gonadotropin, and 47 hours later they have used 0.5 IU of SCG, and oocytes were collected and inseminated with the sperm. Whereas in group 3, it is in vivo fertilization. Transcriptome of fetal brain and placenta were analyzed in 60 plates. Means 6 times they have analyzed the transcriptome for the fetal brain as well as placenta from 3 tissue samples. And they have sacrificed the mice and they have collected the placenta and brain, which can be kept into trizol solution. So this is more of the material, and this is the diagrammatic picture where they have used the plastic disc and glass disc and in vivo fertilization. So now how they have done the calculation. Fetal sex determination was also done. That was done from the tail tip. So RNA preparation, complementary DNA synthesis as done by the microarray hybridization. RNA extraction were done from the 18 samples, 6 from glassware, 6 from plasticware, and 6 from in vivo embryos, and 3 from female and 3 for male. And RNAs and complementary DNA synthesis has been studied by reverse transcripted qPCR. 32 placenta has been studied, where 5 placenta developed from in vivo, 13 from glassware, and 14 from plasticware. So this is the individual mRNA, 22 relevant genes and 2 reporter genes, and they have done studies by compare this gene by comparing from the KEGG encyclopedia. So now this result shows, which I have highlighted with the red ink, that plasticware, the embryos grown into the plasticware culture has 1,121 deregulated genes, 1121. I should also write it down. And in glass, only 200 genes were downregulated. So you can see that in plasticware, how dangerous it is. It is 5.6-fold more genes are deregulated in placenta from the embryo, which is grown into the plastic ware. So that is a threat of the plastic, what we use into the laboratory for the embryo culture. So this is, again, it is a picture where it is show in vivo plasticware and the glassware picture. Now coming to the result, the fertilization rates and embryo development rates and implantation rate remain same in plasticware in the glassware. But as we all know, the implantation rate, if you check in vivo condition, it is statistically significant. Implantation rate in plasticware is 35.6. In glassware, it is 37.3, but in case of in vivo, it is 56.6. So that is in vivo implantation rate for the embryos grown into the in vivo. Now analysis, the differently expressed genes were performed with a threshold of p-value of 0.05 with the student t-test, and 1,121 gene in the plasticware, and from that 621 gene were upregulated, were control of the cellular proliferation, and 493 were the down regulated, where proteins exposed on or secreted from the cell surface. That is the definition of upregulated and the down regulated gene. So whereas in the glassware, when they have used, only 200 gene were detected. Out of that, 111 were upregulated and 89 were downregulated. So now, interestingly, the number of genes that are differently expressed between glass and plastic are much higher, with 434 genes differently expressed. So the plastic is a really threat. Concerning the brain, as it is always the below 49, which is not statistically different, so it is not calculated in this study, and the data was also not shown in this study. So they have only discussed about the placental gene. So they have done the test through ANOVA, and for this calculation of the gene, the clusters of gene, if you see the red, which is kind of used into the plasticware, so that test has done by ANOVA. Up-supervised clustering also is high into the plasticware in comparison to in vivo and the glassware. So what they have done is complete list of modified genes they have compared, and the plastic versus in vivo conducted with the hallmark database. Now, data analysis of GSEA demonstrate a massive over representation of genes involved different type of stress like hypoxic, inflammatory, nutritional, or misconformation of proteins. So deleterious effect of culture on plastics are therefore numerous, and it can alter the gene expression, and it can alter the gene expression involved into the placenta. So this is the different genes and how it has affect the placental act means work. So now in case of females, 547 genes were deregulated after culture on glassware, whereas in plastic it is 1627 genes were deregulated. So that is also again compared with KEGG, which is Kyoto Encyclopedia for Genes and Genome. And when they have calculated for both the gender, female and male, the gene has been expressed in this table, and the gene is responsible for nutritional and protein stress signals. Again, the gene which is regulate the blood coagulation and muscle differentiation can also be affected by this plasticware. The upregulated genes and downregulated genes, which is responsible for adipocyte development and DNA repair, pancreatic beta cells can be also affected. And the gene which is involved in oxidative phosphorylation, like MYC gene, is also altered by this study. So the incubation of mouse embryo onto plastic disease resulted near term in massive alteration on the placental gene expression profile. So the decrease in the functional capacity of placenta, it decreased the maternal fetal exchanges and low birth weight of the offspring at the birth. More gene deregulation in the female placenta than in males, which was somewhat unexpected because generally we feel that female embryos are more robust. So no significant modification of the gene expression profile could be detected into the brain. And ART is a key risk factor for placental disease like preeclampsia or intrauterine growth retardation. So the exposure like bisphenol cannot be ignored because we all know the adverse effect of the bisphenol. And another product which is a cocktail product from the different products such as bisphenol, parabens, and all that cannot be also, we should know that where the plasticware is made up of. So these are all the kind of having threat into the IVF lab. So incubation of human embryos in perform under three potentially aggravating factors. When we are doing the culturing at 37-degree temperature, which may also lead to the toxic compound into plastics or VOC when we are culturing the embryos for 5 to 7 days nowadays in for long. That kind of embryonic genome activation, potentially it is sensitive to environmental changes. The oil what we use for the overlay maybe disrupt the endocrine factors, and this disrupt can also had the effect on the pregnancy and implantation of the embryo. So according to this data, glassware culture appears closer to in vivo. So that is what is about. Thank you.
Wonderful presentation, Charu ma'am. I mean, you have put in so much of effort and explained. I read the papers. They are really, coming out and deciphering it. You have put it in a very nice manner. When I read these papers, I was first of all flabbergasted that what is happening. I mean, should we use plastics and should we shift to glass? But the more I thought about it, I had a lot of questions. So sometimes I would like to play the devil's advocate here and go against what is going on. Both papers are quite interrelated. Let me begin with the first paper, that is, we are talking about the consumables. And I think that it is basically talking about the sperm preparation that we are using for testing all the other consumables. My first question arises, this sperm preparation that was actually as a controlled use was made in a plastic container. So maybe the phthalates, maybe the bisphenols and plastic wares, they actually came from the control itself. So that is not discussed in the paper. So
No,
the
that was it was yeah, that was
yeah,
not discussed in the paper,
exactly.
but it is written that the control has been individually tested for the toxicity, and then it
Yes,
has been considered as a control. But yes, that was a limitation of the
yeah,
study.
right. So the follow-up question to that is, all these are experimental. This whole experiment is done regarding 30 minutes usage and accumulation of all these 17 different consumables, 8 or 9, whatever you want to call it, different kinds of accumulation of consumables. Now the question is that when we use these consumables in real life IVF cases, their exposure is not that much. So 30 minutes, yes, except for the dishes. Actually, if you have wonderfully put in your presentation that whether it's the ICSI dish or your plain dish or your culture dish, whatever it is, they do not have it. I think 8 or 9% is the thing that you have found out. So when it comes to exposure on your regular things like the dishes, there's no problem. But even then, the sample that we prepare, it is prepared and used immediately, so exposure is less. So real life results may not actually be the same that we have what they have displayed. That is the second part I disagree with. What do you like to comment on that?
No, yeah, partially you are right. Partially, what happened, as you said, that we minimize the use when transferring from, use whenever we are using kind of transferring pipette or dish or something.
Yeah.
The things, whatever we are using, like when you are using flexiplate, that is also made up of different kind of material, and it is more toxic when you are using for denuding and all that. The time sometimes it may vary depend upon the operators, but for the dishes and the sperm preparation which we keeps more than half an hour, you got my point. So few
Yeah.
combinations is used more time, that few we are using for the very less time.
Yeah.
No, so and particularly the dish preparation, when I was going for this study, I saw that what is the made up of material. Then I came to know that flexipate is actually made up of the some other material which are more toxic compared to the dishes and the tubes.
But that is also the exposure time they have mentioned is 30 minimum 30 minutes.
Minimum 30 minutes,
Yeah,
correct.
so when we are actually manipulating the oocytes, embryo, whatever, it's
Yeah,
hardly for a few seconds, not even
yeah,
minutes. So
yeah,
the
absolutely.
comparison should be that. That is,
Yeah.
I mean, it's wonderful that I think I even got scared. But when we do have a reality check and we think in realistic values and time, the exposure is not that much. So do you need to be as much scared as the paper is trying to tell us?
Yeah, the limitation of study is everywhere, it is there, but one should be cautious for, kind of this will give us, kind of checklist, to make a checklist for ourselves. It
Right.
may be that dangerous as you said, but the threat is there. So being a senior embryologist or embryologist working in this field, we can, make a new kind of SOP where can be minimize the use of disposables, and every person in the lab should be aware about uh, the material which comes in, whether it is tested or not.
Yeah.
Because some kind of purchase manager, to save even the 1,000 rupees, they'll order from the different company.
Very true. So I think what they have mentioned is your CE marking, your MEA testing, and SSMA. These three tests. I think we should go in for products which have been at least okay. The HSMA is something which is done separately as an experimental one.
Yeah,
But maybe a CE mark in MEA is something which you should check in all your products. I think that will be a
absolutely.
good cutoff and good maybe a sign check
Take
for
home message, yes.
take home message. One more thing about the paper I understand is that
Nishad, just
they
to
have not. Yeah,
interrupt on the same thing before you go ahead. I think it's what you have raised is a very valid point. We are not going to put in anything for 30 minutes. I mean, Flexipet, even if it is very toxic, we are not going to put in for 30 minutes, as you said, except for the dishes. But the question is, it does raise an interesting thing and an interesting study because, I mean, we can repeat it ourselves.
yeah,
I mean, we have about 50 people out here. And if one or two get together and say, okay, let's see what we are exposing and let's do it. Because they've
yeah.
just done a sperm motility assay. So it's no big deal, doesn't require any funding. I mean, maybe people here can start doing it.
Yeah. A follow-up question, Rajvi ma'am, I would like you to also get involved is they have not mentioned about off-gassing of all these
Correct.
disposables. So
Yeah.
I was coming to that because see, it's not the microplastic, but rather the bisphenol A, the phthalates, and basically the stearates. These are the things that are responsible. If you are off-gassing for maybe 2-3 days, so the leaching is stopped and it's, I mean, it takes place and it's already evaporated. So
Yes.
that is one control. Maybe I'm not even saying that this is a wonderful paper. It has
Yeah.
literally opened my eyes that we should be careful, but maybe a lot of more research is required
Absolutely.
that you take care. And second, this is mice. Only 50% of your DNA is matching. Even if you say chimpanzee where 99% is matching, but that 1% difference is what makes us human and the chimpanzee a chimpanzee. So
Are you
unless it is
talking to
done in
the
human
about the
models.
second paper, the gene
Yeah,
expression?
gene
Yeah.
expression.
I don't know, Nishad, whether you recollect, but when we made our own culture media ages ago, you
Yeah.
obviously don't belong to that generation.
Oh no,
The
I have done a lot.
Okay. So the fact remains that when they prepared culture media and they prepared culture media from tap water, and let's not discuss tap water in Mumbai or tap water in UK or tap water wherever, the mice embryos survived that. So, if there is, and we don't know what this gene expression actually, but the low birth weight is a fact. I mean, we all
Yes.
know that IVF babies are low birth weight.
Right.
And I always believe that IVF babies are possibly the pregnancies are most pampered in general, as far as
We do call them a precious
yeah,
pregnancies.
But
precious
that,
pregnancies.
but yeah, low birth weight is not only because of the lab factors, no, the clinical
Many
factors
factors.
and post conception, how that lady has been, taken care, her exposure into the real kind of toxic materials in the
Right.
environment.
No doubt that it's not
Yeah,
that millions of babies have been born and healthy
correct.
babies. So it's not to create a paranoia, but the fact remains that we need to keep our eyes open. I mean, it's so
Absolutely,
much of gene
absolutely,
expression alteration. Yes, yeah,
absolutely.
go ahead. You wanted to discuss something else, and Sarguna
No,
also.
I think yeah. So obviously off gassing we have covered. We have covered the parameters. Plus, obviously, real life conditions are very different from the experimental ones. So that is one point of discussion I would like to put in that changes the whole game altogether. We do not put that much amount of time in the consumables. The embryos are not that long staying in any particular consumer except for your dishes. So that is one thing. And I think it's okay. Can we shift like from the other paper also? Yeah. Okay.
See,
So
Dr. Sarguna wants to say something on the first paper. Yeah.
Yeah. So obviously, absolutely, they have what Nishad was talking is absolutely fine. This is the query everyone would have on reading this paper. But sperm toxicity testing, in the olden days, probably I have not done the whole batch sperm toxicity testing before a batch IVF started, but my mentors have been doing it. And I was the one who was exposed to sperm toxicity testing where we were suspecting some kind of toxicity from a particular consumable or a disposable. So that's how I learned that technique. But then I knew that at the same time, like 20 years ago, before the batch started, every consumable which was supposed to be used in the batch was tested using the sperm motility assay. So that is how they have been doing. Now, at the end of this discussion, probably we may have to go back to these points, like, are we going to do anything like that ourselves, kind of. Why this point comes is like, there is a heterogeneity in the study anyway. Whoever is trying to do a study, you are not going to have millions and millions of sperm from one person so that we have a uniformity in that test. So obviously they have had different sperm, different mice, different strains, different timings, different variables. So there was a big difference in everything. But at the end of the day, they were just finding something and they have given clearly the indication of as a limitation, actually. So this is the point I wanted to say. Second one was the CE marking. So what is testing really the toxicity of the materials going to be used by the manufacturers is the guidelines, not any regulation we have. One is MEA, other one is sperm toxicity testing. Talking about the CE marking is not the quality indicator, I would say, because CE marking is something that is only applied in the Europe, where medical devices coming from any country internationally, they can deliver them, just have a business with EU countries. So this is more about business, not about the quality, comparing to our MEA and sperm survival assay. These are very direct. So anybody, any manufacturer dealing with the disposables are doing the MEA testing as well as the sperm toxicity testing, as we all know. And as Nishad mentioned, the off-gassing was absolutely a great idea. And many of my seniors have always mentioned that you have to do it just to get rid of the toxicity to a certain amount. But again, like how much we can just stick on to, well, okay, again, going back to the papers, articles about the off-gassing, then they compared 0 hours, 0 days, 7 days, 3 days, 4 days, whatever. And then they were finding differences in that. And how long we are going to give the off-gassing time for. Is it going to be hours? It's going to be overnight? Is it going to be 2 to 3 days? And do you have enough space in your laboratory? Do you have the aseptic conditions? Are you not worried about the contamination at the end of the day? So there are a lot of things. So what we were following in one center, Alex was the one who was supposed to be just giving us some ideas about off-gassing there, was few hours. So that is one thing which was like, in a laboratory routine, it was easier for us rather than keeping them forever. So few hours it was off-gassed, and then we started going for probably like 3 hours or something. So we were trying something. Whenever we had some issues with the results, that's where we are going on. We were not doing a direct toxicity testing onto the embryos. Actually, that is one another point I wanted to really say. And so what the other part is like the guidelines. That's what only the guidelines we have no regulations. ISHRE, for example, the laboratory practices, recommendations for good laboratory practices, simply putting a couple of lines about these disposables and media is like, you have to really test they are certified, that they have tested by the manufacturers. That's all. And then you have to really follow how they have like packed and dispatched. And these are the only points we have, but we have to really still follow it. And how many of times we are checking whether everything is being supplied with the certificate of analysis. So do we have a certificate for all the products? Is everything just done by the manufacturers? Because they were surveys, and then they were trying to talk to many of the manufacturers, and some of them didn't want to disclose what they had done, how they did the quality checks for the consumables. And some of them had disclosed. There is a variability again. Yeah. So that is another point I wanted to really tell. Otherwise, sperm toxicity testing is one thing what we can do in the laboratory right away if you have a query. That is the only stuff I can see. But it may be having some effect. So we may have to look for more papers, as Nishad was already warranting.
So Sarguna, in this case, when I have started, IVF way back in 1999, we used to do conventional IVF. ICSI were only for the indicated cases, irrespective of like ICSI for all. So to have a check that fertilization rate, we used to check for every patient after when we, on the second day at the fertilization check, we used to keep that dish for 96 hours for the sperm survival test. So that we can see that fertilization rate is because of the sperm or because of the oocyte. So it is kind of two tests at one go, you know. So
yeah. This is the egg factor and the sperm factor.
Yeah.
Hello. May I ask a question? Actually,
Yeah,
two or
Sonal,
three.
go
Yeah,
ahead.
please, please go ahead.
Go ahead.
So in the second paper, I just wanted to know that the glassware that was used, how was it sterilized? First thing. And whether the down-regulated genes that were seen in a glassware or in a plasticware, were they the same or were they the different genes that were down-regulated? That was my another question.
Yeah.
And my third question was, we have a lot of other plasticware, like, you know, the goblets or the Viso tubes that we use. Although we know that the leaching of the plastic will occur, more so when the temperature is high, and in cryo devices, it should be a little lower, the rate of, leaching. But when we are storing our cryo devices in these goblets or Viso tubes, we usually do not change those goblets or Viso tubes. So all we do is we audit, annually, but we don't happen to change those goblets. So does the expiry of such plasticware, in which there is no direct contact of the samples with the plasticware, will the expiry also happen to, have an effect? Has anybody studied about it?
So Sonal, first of all, it's a very good question. Thanks for asking that question. The question number one of your, that the glassware, whatever they have used for this study, how they have sterilized. So the paper did not mention that the glassware, but it is a product which is ready-made available, and probably the manufacturer has, kind of pre-sterilized and that is used. It was not exceptionally sterilized into the laboratory for this particular experiment. So that is the answer for question number one. Coming to your another question, that whether the down-regulated gene in a glassware or same in plasticware or not, so that are a different set of, kind of few are same and few are different, but that is another that we have to study the paper in depth in compare with the encyclopedia where they have compared. And the third question of yours is a very, very interesting questions. So Rajvi, would you like to answer or I can go ahead?
Go ahead.
Yeah.
Go ahead.
So the plasticware or the tubes, whatever we are used for freezing the, male gamete semen sample or embryos. So, the study I have not come across, but definitely when we keep the embryos for 10 years long and the baby were born after 25 years of, frozen embryo, it itself indicate that, embryos are not affected. Another, coming to the point that when we are freezing the semen sample and when we are warming or thawing the semen sample, it is not only the cryo vessel or cryo vial which is responsible, but the technique for freezing, the cryoprotectant, and everything here matters. Not only the use of the plasticware what we are using. So that's what is my answer. Rajvi, would you like to add something else?
I think these are some of the very early studies that have come in. So there is not much literature, but it surely opens our eyes, and it goes way, way back in time. Earlier, we never bothered about VOCs. And it was only Jacques Cohen's paper report, way back in the early 90s, where he talked about various levels of VOCs, and then the CODA came in, etc. Now, there is one report where in the box incubator, there's a high levels of styrene. And now, where does that come from? It obviously comes from the plastic dishes. And I'm sure we have improvised on the dishes over the period of time. So the big thing that, and there's somebody else also in the audience who's asked, what about the cryo devices? So I don't think anybody has an answer to that now. I mean, we can only presume that it's all safe. But in science, you don't go on presumption. I guess somewhere one will need to experiment. Secondly, gene expression studies can be very confusing. I'm not a geneticist, but I know gene expression is very dynamic. So whether, yeah, we can, go to get paranoid or get worried, but yes, it's altered, and we need to keep our eyes open, over a period of time, whether something can be improvised or something. So one thing that comes up, two things that come up, is that the age-old human sperm motility assay is not a big deal. Anyway, we are processing sperms all the time. So it can be easily incorporated in any lab. It doesn't require, yeah, in terms of cost, etc., but it's trivial. So I think that's something which can be considered whenever you're adding something new to your system. So, it may not reflect what happens to the embryos, but it's something that can be added. And another important factor that comes in is the cumulative effect. See, individual dish, individual plasticware did not have cumulative, yes. And we are doing a cumulative thing. So I think all the studies should actually, like Nishad said, we need to do a complete analysis in a real-time situation. So that can be good with the sperm. Yeah, Sarguna, you wanted to add on to this?
I just would like to add a couple more points about
Sanket,
the glassware. Sonal's question about the glassware. And in fact, in this study, it is not clearly given what kind of sterilization they had done. But we use the glass partial pipettes for sperm preparation. And in fact, first 10 years.
Needles are glass.
Yeah, need glass. And then we used to pull glass pipettes for everything, all the manipulation, for denudation, for movement and everything for the first 10 years. Now I have stripper. And stripper is having bisphenol A. Now they changed to bisphenol S. And they say it is very toxic. And then I don't mind going back to glass pipettes. But now how the current generation is like. This is
Again, we have to talk about the off-gassing here. If you keep it open for a while, it does happen. Like, come on.
Yeah.
And this is so like any glass items coming to us with the sterilization, probably they would have applied that sterile products available for the study also. This is my guess. And second point is about the toxicity. So cumulative toxicity, not only from different kind of consumables, but what is really happening in embryos, growing embryos, is already they are cultured in vitro into a new environment. Laboratory conditions are different, not like in vivo. So they are already under stress. There are multiple factors. This is multifactorial. A lot of stress onto the embryos. So studies are showing, the literature evidence I'm saying, like even if a particular chemical is showing a low amount of toxicity, might amplify its toxicity when there is an added stress for the embryo. So this is how this toxicity is affected. And what the embryo needs to do is it has to really use different defense mechanisms, which it doesn't have by nature in vivo. And they have to use multiple sources and use a lot of energy to fight against it so that they get depleted. And we have compromised embryo development, which could cause the gene expression patterns, alterations, or any aberrations. Everything is related to these mechanisms of energy loss. This is what I was just thinking, just to say.
Sanket, you wanted to add something?
Yes, ma'am. Yeah, well, firstly, I would like to thank all of you for getting this interesting discussion. And yeah, what I found was one of the paper in 2016, which read about basically they stated about this impact of this BP and the IVF embryos. So what they found out was neither the culture media or the protein supplements which has been used contain high detectable levels of BPA. Right, but however, they found out that whatever the strippers, as Charu ma'am said, the strippers what we use do release certain amounts of BPA into the culture environment. But the point is, this was very interesting point for me when I read this paper. So they found out that whatever this disposables that releasing this BPA into the environment, those levels were much lesser when compared to the BPA levels of human serum as well as follicular fluids. That was about 2 nanograms per ml. That means these were 0.5, that's all.
Yeah, I agree. I read that paper. Yeah. So that's why I said that the BPA comes from the other sources are more, kind of affecting the system.
Right. Because it has become...
Gatimel et al. 2016.
Yeah.
Right. So, it has become integral part. We are blaming the dishes, strippers and all. So follicular fluid में क्या करेंगे हम लोग? मतलब we cannot avoid it, right?
Sanket, can I add one more thing? The paper, the first paper which Charu ma'am was mentioning about the consumables. So I made a list of things that are actually toxic. I mean, it's a accumulation, right? But out of that accumulation, what is exactly affecting the most is just a moment. It is basically
Thalet?
one is the flask, which we don't use nowadays because
Hmm.
what like how Rajvi ma'am mentioned that we used to make our own media. So
Right.
those flasks were used. So that is one. Second is there were 2 lots of your cement containers, cups. But one lot which is actually MEA tested, that did not have it. So that is how we can actually safeguard and use such things. That is that is the second thing. And I think third thing is, where is it?
Plus lid and cap, we do not, kind of any time
Yeah.
touch the gamete.
Yeah, exactly. So these are the things that will make a difference. Otherwise, if you use it, yeah. And the third is the syringe, in which we are maybe loading the embryo or even sample checking, that has rubber inside. So obviously that is toxic. Here we are not allowing anything to come in contact in general. So these are the 3 main crucial toxic substances which we in real life do not use that much as it
So
is mentioned.
I have a question here about the storage. Sometimes the oil comes in plastic bottles and we store it in the refrigerator. It's not always the media comes in bottles.
Yeah, so
As you
now
said,
those
olden
bottles are
days might have
actually,
been glass flasks probably, but
yeah.
now we may be having. So we don't know about the effects of it
So
during the
the plastic
transport and
bottles
Still,
nowadays
storage.
use are made of
Vitrolife does
Triton.
provide with glass bottles only, even to date.
No, but many times you do get your plastic bottles also. But now if you
Oil
ask,
particularly comes in.
Oil.
yeah,
Yeah.
manufacturers also, na, they are made of Triton, and Triton is not having BPA, not
Exactly.
BPA also.
Yeah.
So it is even safer. Your water, your media, your oil is made of Triton. That is not having all these things. So if you talk to the manufacturer or ask particular brands what they are using, you might be more careful and then decide what media, what oil, what other things you want to buy or use.
And generally we order as per our need. We don't
Yeah.
store in a.
See, and
Sure.
temperature is another factor. If you are storing at 2 to 8, nothing going to leach. Temperature is the main factor. So if you are keeping it 37 degrees, then it is going to leach. And if you off-gas it for a longer time, it is not going to affect it that much as it is mentioned in the paper. Yes, it does have an effect, but in real life, situations are quite different from
Might be
what is
different.
actually mentioned in the paper. Yes, it is an eye-opener, but we have to be realistic also about it.
So I would like to add a particular paper. I will share the information from the paper that has been from the last year. Alpha scientists and then International IVF Initiative, all the scientists put together, they have published something called Sustainability in IVF. That is a recommendation by expert panel. So there they were really talking about carbon footprint and how all the disposables are affecting the environment and the human beings, and how we are going to have a control over laboratory parameters and everything. So I would like to share some information because they have openly mentioned about the manufacturers' names there, and that what changes they are bringing in to minimize the toxicity, whatever is there with the consumables. It's like, first one is Coring and Thermo Fisher Scientific have developed more environment-friendly laboratory consumables with the lower plastic content. This is one point. Other one is Vitrolife has announced the development of bioplastic. Bioplastic is manufactured from the bio-based polymers, more like natural, so that you will have less effects of embryo toxicity. And other companies like Cook Medical, Cooper Surgical, and Thermo Fisher, again, they have stopped using certain chemicals in their manufacturing of the plastic wares so that the effects are going to be, so they found something alternative. Even for packaging, they're going on paper-based ones rather than plastic-based. So this much of effort has been put in through from the manufacturers that they are doing something about it. Also, the scientists are trying to put in what else can be done in the meetings, and they're publishing things. So for me, this is one thing, what the manufacturers are doing. What we are going to do, this is one. And what can happen in the future is like proper regulation on all these things. It may take ages, but we have to still have it. That is what, some improvement in the regulations. And how many of you want to go back to anything like, in case if you think like plastics are still giving higher embryo toxicity than the glassware, at least partly, would you like to shift back? We are having a few like fascia pipettes and ICSI needles, as Nishad correctly mentioned. Would you like to go back to pipette pulling or something you want to do about it? This is one. And or are we just looking for something, some alternative? Like there was one called Diamond Dish, where Sommer et al., Germany, I think they have found something. They made up quartz. It's made of quartz, and they have micrometer thickness of diamond coated. It's very expensive, but they were claiming that these were having very less toxicity and no retaining of our voice, etc. Blah blah blah. But it is very expensive. That's one thing. So are we going to find out something really cost effective so that we can have some kind of alternative if it is proven to have higher toxicity rate from this? This is the question I had at the end of it. Too many questions, a lot of ambiguity. So I would like to listen to you all.
Yeah, so what happened, like whatever your suggestion is very true, but in day-to-day lab practice, pulling out pasture pipette may not be possible, and that also needs an expertise. You know, every cannot do with the lamp and all that, and if it is done, there is another added heating of that lamp and all that. So we have to deal with whatever commercially available. That's what I feel. But we can, be a little more skillful in time management for handling all that, and we can be little cautious for this product. Plus whatever we available, like as you said, that manufacturers are aware of this, they are also putting lots of effort in manufacturing the dishes from the natural sources or the bio dishes. That is not that costly, so we should see the material what comes in that has been tested or not.
Yeah, just to extend what Dr. Sarguna Devi said. See, the point is, ma'am, Charu ma'am, the point is, if you go to any infant shops, you get to see the label, right? BPA-free toys and the water bottles or everything. So when you're getting that for the cheaper rights, why don't you make dishes based on that? What are the material they're using? I think it sounds logical, right?
So that is Sanket that we wish to have, but even in the for same kind of media, few things we do not know which is available commercially, that what are the additives in the media that they are using. So that is manufacturers what kind of secrets that they don't want to disclose for in this competitive world.
And also we have to think about sensible use of consumption.
Yes,
so you waste
absolutely.
only minimal. I have
Absolutely.
worked in different laboratories. In some laboratories, it's always 1 tube per follicle and 1 dish per tube. And it's
Yes.
too many wastage waste. And here I'm just saying like 1 or 2 tubes are rotated, and then we are just having minimal amount of dishes used during the processes.
Absolutely.
So that is I think very important that we have
That is wise. Yeah.
sensible about it. Yeah.
So somehow I feel we need not really, I mean, yes, what is the contribution of IVF to the human population? Minuscule. And, we are contributing to the environment negatively with all the disposables, but it's very, very minuscule compared to what we are using as humans. So sometimes I find, especially in larger organizations, that, minimize the use of disposables. But sometimes it's at the cost of, you may be losing oocytes in the process. So I do feel that we need to be a little balanced in that phase. The thing is, I am going back to glass, having used that. We used to get ready-made glass pipettes. The next question is, how do you sterilize them? You autoclave. Then the next question is, what is the water used in the autoclave? It enters into the, so we get into a loop. So I can't even imagine or dream of going back to glass, although it seems much more safer. So we still, I think there's lots that needs to be, these papers were primarily eye openers, that somebody's thought about it. Nothing works in isolation, and I think that's the biggest message for science. Because for a long period of time, and to give an analogy, people have always looked at haldi in a good way all over India. It's something, but when you look at curcumin, the active principle, they don't find it effective, but together it's effective. So the biggest message to me is the cumulative studies. Of course, they are not really relevant in the real situation. 30-minute exposure, look at the lid of the dish or lid of the tube. But I think it's an eye-opener, and maybe the large group of embryologists that we have in India, small studies can be done to see whether it's really making a difference. A motility assay or stuff like that. And yes, we are at the mercy of the manufacturers. What is available, we use. But remember that we are also a large population in that sense. So if we demand certain things, they're going to give it to us. So, both ways, it's a two-way dialogue, and it's a good thing. So, I mean, I feel a big concern is coming from the use of the cryo devices. A couple of people have asked that question, and I don't think anybody really has an answer. Whether storage at low temperatures, whether it's going to affect, I don't see any reason, but you need some chemist here to tell us what exactly is happening. If there's some leaching happening, and whether it really would it affect. Because see, most of these things will be organic compounds and they'll not find their way in the culture medium. But possibly these plastics are leaching, and because of the oil, they accumulate there. So even that's another, apart from the degassing, off-gassing part that we discussed quite extensively. Because if you're not off-gassing enough, that's just the cause. You're blaming the plastic, but it's the sterilization process. And the second thing would be, the leaching that could be. So maybe it's just accumulating in the oil over a period. So I think we've had if anybody wishes to add anything, audience, please feel free. I don't think anybody has answers to anything here, but it's a cumulative thought which may help us look for answers. So it's open to all the audience present there. Please feel free to unmute yourself and speak if you have something to share or an idea. Otherwise, we can call it a day. So unless my panelists have any last word to say. So,
just relating to the placental gene expression, I had my reservations on certain grounds that yes, I completely agree that yes, the study is robust, no doubt. P-values are amazing, analytics and data is superb. But I have certain doubts regarding that paper. First of all, it's not a real life scenario because it's done in murine models, that is your mouse models. So that is one factor. Second is super stimulation, that hyperstimulation of the patient is given. So these are the factors they might also affect. Glass,
And
yes,
another limitation, what I have studied in this paper is the mice use for production of oocyte. That is a different breed. And what I read that the transfer into the different breed of the mice. So that also kind of differs.
right. So I mean, yes, it is something, but you cannot say it. I mean, pinpoint that this is the only factor. But
Yes,
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 I completely agree. We have to worry about it. We have to make
yes.
sure that we reduce our plastic footprint, or at least take care of the stuff that we are buying, like going for off-gassing, and use them very sensibly. No doubt
Yes,
about that.
absolutely.
Nishad, just to add point to what you said, see, first of all, I don't understand how these pathways are made. See, all the genes and even the mRNA levels in the cell, they're very spatiotemporal expression. They show spatiotemporal expression, and they're comparing with cake pathways. Cake pathways made out of whatever the animal models, and they're comparing with this of humans. I don't see any logic over there.
That is exactly, see, that's why I said chimpanzee 1%. You compare in human directly, but this is 50% different DNA altogether.
And
So,
they do have different expression, levels of expression.
Yeah.
Even see, if you look at the temperature,
in humans, it will be totally different. You can't just, I mean,
So
pinpoint.
the thing is, like, probably we have to correlate things like how much of a maternal exposure to plasticware, for example, food packaging and then bottled water they have, even with the whatever they have at home, the kids' toys or whatever, and then they found the particular chemicals present in their blood and everything. They have seen the translocation of that to the placenta, as well as there was an effect onto the fetus. This is
Let me throw a curveball on it.
No, yeah. So when they are talking about, they could find that, and then they are affecting, that is the reason why they have lower birth rate. This is what, as they have said in the paper. So correlating that, there may be some effect, but then how much, as you know, like as you discussed, we don't know. That is the only thing. There may be effect, and overall, we may have to really think about like less plastic usage. As you
See, one more thing that factor that I believe is the person itself. Let us talk about human models. Like you mentioned, we are already having so much of microplastics. Is that affecting the oocyte and the sperm quality? And is that in turn affecting the placenta? That factor cannot be ruled out.
Nishad, anyway, there's so much of literature of the role of plastics in declining sperm count.
Yes.
Now the whole thing is how much are the environment and individuals contributing?
Yes.
Because you are talking so much and it shows affecting hormones and especially pre-puberty, etc. Then first thing is that plastic milk bottle should be banned.
Yeah.
Right. See,
That's
my
the
point
first
here
thing which
to
can
say,
be done in a big
The
way.
couple undergoing IVF treatment themselves are exposed so many things in before or during the treatment that cannot be ignored. No.
No, anyway,
so
we would like to put in our
Yes.
bit also in whatever
Right.
best way.
Absolutely.
I think Dr. Ravi wanted to say something. Go ahead.
Yes, madam. It was a very fantastic discussion about the use of disposables in our day-to-day routine culture systems. So I just wanted to know if anybody has studied or is there any solution that we can avoid and go ahead with, you know, a proper culture condition, whatever the existing.
We didn't get your question, Ravi.
No, we are discussing about the the toxic effects of all these
Yes.
disposables in our day-to-day culture system.
Correct.
Is there any solution? Like, okay, we can modify like this and we can get a better result. There are some tips and tricks. Is there anybody has studied or anything is available?
I don't think anything is available. I mean, and even if anything we suggest, that would not be scientifically valid because
Yeah.
nobody has
Yeah.
any data.
Exactly.
nobody's tested it
But
actually. You can just say.
nobody's tested it. The comfortable thing would be to say 30, 40 years we have been using it. You know, it started with test tubes. That's how the test tube baby came in, and we've shifted to dishes.
That's
And
fine.
nothing dramatic. But I think
Yeah.
as scientists, we need to keep our eyes open, ears open, brains.
We
and
should be
think about it.
cautious.
Yeah, that's fine. That's fine. That's fine. I
Yeah.
just wanted to
No,
So,
there's nothing.
right now,
In fact,
right now,
that made it such an
Madam,
interesting paper.
right now we are doing the home pickup and using the screening dish, big or whatever the open dish is. So once if you pour the follicular fluid into the screening dish, so can we use it continuously instead of using every new dishes? So what is the once if it is almost rinsed, suppose if you use one follicular fluid, one dish, and just you throw it. Of course, we are practicing using of multiple times in the same dishes. Or many people did it. No, it cannot be. You just take some 5, 10 dishes all over the laminar flow and put one. So is there like using a new dishes and using the you use dishes? So
No,
what is your
until and unless is this you're clear, and Ravi, I don't think because of the plastic threat you should change the dish.
Yeah.
If the follicular fluid is having a bloody and kind of more of RBCs and the dish is not clear, then in that case you can change. Otherwise, you can use the same dish for the screening of the eggs.
Because if we presume that it is
I'm just
because
asking for
of
any practical tip.
no,
See,
what
Dr.
you're
Ravi.
saying about supposing we say this effect is because of not sufficient off gassing, and if we're going to use more dishes, we are going to contribute more negatively. Yes, using less dishes would, in a way, in a minuscule way, contribute by less plastics thrown in the environment.
Exactly,
So,
madam.
it would contribute. So I guess we take a balanced view that as long as things are visible, we continue using it. When things dry up, or as she said, it's blood stained, visibility is poor, we use more. So I guess
See,
it
and
will be what
we also have to understand one more thing is the exposure time. If we just consider these two papers, they are saying 30 minutes exposure time. Are we using those dishes? Hardly for few minutes, few seconds. So you can reuse your dishes. I mean, logically, it doesn't make sense for using so many dishes. Even the paper is saying 30 minutes, and then it's going to, if you see the cutoff, it's not 0.85. Most of the things are 0.77 or 7.4. So it's not even like drastic effect that is causing. So that is
less
toxicity
than
test.
few
Yeah.
seconds in the dish.
Yeah.
that-
And those things which are affecting are not dishes, first of all. Those are your syringes, those are your flask, which we don't use. These are
Transfer
two things.
pipettes, yeah.
And semen cups. So when we talk about dishes, it's not that harsh on the when it comes to the use of microplastics and the phthalates and the bisphenols. It's mostly these three things that we have to consider out of that paper.
I wonder the practice a lot of people have is using the syringes to aspirate medium, etc. I've been more comfortable with pipettes. So any comments on that? Instead of using syringes to aspirate the semen sample, etc. The normal syringes. That I mean,
Many
that's in
a
terms
times,
of
many a
tips.
times I directly pour it from the bottle to the like tubes.
Yeah.
Even I don't use the pipettes also.
Yeah.
It is
No,
about the
that is, yeah,
sterility.
it is, yeah.
Yeah, this is about the
Except the-
sterility and who is preparing the media and how
Yeah.
much of competency they have. That's how we design the
Absolutely.
protocols
So
accordingly.
I've personally
And-
never been, maybe not logical or illogical, using syringes for other activities.
Yeah.
So that's a small tip, Ravi, if you can consider it.
And
Yeah,
getting back to Nishad's point about the sperm 30 minutes of exposure, the traditional sperm toxicity testing doesn't ask you to just expose it for 30 minutes. You are just trying to have a contact for just a minute. That's all. And then you are putting it back and then going to check the motility in 30 minutes, 24 hours, 48 hours. This is what we have been doing. It was never like we had to expose them for really long to just check the toxicity. If it is toxic, it is toxic. Just in a minute.
Well,
That's
let us
how-
not make plastic the complete villain. I mean, there are so like
It is a
we always
partial
say,
villain.
it's like if something goes wrong, blame the media, blame the embryologist. We are just talking the same. Don't blame the plastic.
Embryologist.
There might be other reasons.
It's a partial contributor. Somebody is
Yeah.
worried if you're going to use the same dish again and again, Ashwini, would the temperature change? You're saying reuse it for screen. Not
Screening,
really.
yeah. It's like I've
See,
used
if you
both
have
the
done the
ways.
off-gassing,
Yeah.
if you have done the off-gassing, that is the leaching and evaporation of these stuff have already
No, she's
happened.
worried about the temperature, but it's on a heated state. So I don't
Yeah.
think temperature would be an issue. Yeah, Ram Prakash, you wanted to say something?
Good evening.
Yeah,
Yes,
go ahead,
yes. Hi.
go ahead.
So first of all, beautiful presentation, Dr. Charu, and great discussions are going on. So that's why I had to jump in. So basically, I just want to share experience. Recently, I was speaking to one of the manufacturer. So they have shown me like we are manufacturing these dishes and this. So we just randomly, I mean, like I'm sure you all have heard about Green IVF initiative, and where a lot of people have started, reducing the use of dishes by reusing them. So it's a good idea to reuse them, as like there were many centers they are still practicing that they are not reusing the same dish. So again, reusing will reduce the plastic footprint. But if you see our IVF industry all over India, I mean, what the plastic waste we generate is the 1% of what Delhi AIMS generates. So we have to remember that also. So we are not contributing a lot to, we are not destroying environment much, because the material also which we get, PP and PS, polystyrene and all, so they are easily recyclable. And there are a lot of, most of our waste segregators, they take dishes from us. Only the dishes which has blood on it, they, don't reuse them. Otherwise, rest of all the plastic dishes, they recycle it. They use it for other purposes, maybe for the sheets, for other plastic, for other plastic materials and all. Only PET or the plastic which are not recyclable or not easy to recycle is the PET and the higher grade of that. So I think we are not contributing as as other branches and other things. So we can be little relaxed on that, and of course we can reuse them to save the footprint or carbon footprint.
So yeah, it is very interesting, Ram, and it gives me another thought that whenever we are disposing these things, like like now in our household, in many like metro cities, we have a different dustbins. In that case, also some company should come up to take this, particularly petri dish and plastiwares, whatever we are using, to the recycling or to the kind of how they are doing, because nowadays only one company which takes all the all the materials from the center, which includes gloves, which includes everything. But instead of that, if we also segregate, I think that will be also more interesting if can.
So see, the companies, biomedical companies, they always tell us to segregate. But our juniors, or as a
no, that is only sharp materials which they ask us to keep separate.
No, I think for BME they have a separate for the plastic. They have separate for the blood.
They have separate for the glasswares and plastics, and even for the
separate for the vials,
medical waste management.
Broken glass. So again, we have to be as a lab director, so as a senior embryologist, as an embryologist, as a junior embryologist, we all have to be responsible to discard
Absolutely.
that.
But Ram, unfortunately, it's a very good idea. But in reality, all these things are medical waste, goes in a dustbin, and they go outside and everything is burnt.
Done. Yes,
That's
absolutely.
the true reality in India
So if
and
you are
in most of the
giving your
world.
waste to a biomedical agency, they are not doing this.
No, even biomedical agency
They do it,
for
they do
that
it.
matter.
I have checked it. In Nagpur, it happens. everything is taken together, बोलते हैं, करते कुछ और है. It goes in the dump.
It's not
Actually,
about that,
it's,
Nishad. The point
from,
is, if you
yeah.
want to get for NABH accreditation or any inspection is there, they check for this BMW. At least for that, you have to dispose in a right container.
No, we have different.
No, we
companies.
are disposing in
take
a
it.
different
The person who
container.
takes it away does not take
Forget
it to
about the
that.
endpoint.
Okay, so
No,
I'll
just
intervene.
just one last thing I would like to
Yeah, go
share.
ahead.
All this discussion remembered me my master's class, where my professor walked in the class and she wrote silent springs. She asked us to explain what is silent springs. Like we were lost. What is silent spring? Then we slowly realized like it was a developmental biology class, and she was taking something related to fecundity of these birds. So we realized during the World War II how this indiscriminate use of this chemicals and pesticides caused reduction in the fecundity of certain birds in certain islands. So we are not that far with the use of this PPA and phthalates as of now. Of course, we have to take this with pinch of salt and we have to minimize the risk as such as possible. But of course, yeah, we have to remember that.
Yeah. So I think we've had a very interesting presentation, thanks to Charu, and a very nice discussion. It's given a lot of thoughts. We deviated a bit to a very important topic of disposing our, the discussion on how we are going to manage the biomedical waste. See, when you are in a large hospital, things are different, everything is set in place. But when you have independent clinics, how is this biomedical waste handled? So I think it's a good topic of discussion. How do we generate practices? Because we are so openly saying, it's all there on paper. Maybe somebody comes for inspection, we follow it. But, finally everything is just accumulated and burnt. So I think it's a good topic for subsequent discussion. And I would ask people to volunteer. We'll have a panel on actual biomedical waste discussion because people seem quite passionate about it, and it's important on how we handle this. So thank you very much.
And
Special
in
thanks.
next-
Yes.
Thank you.
Madam, in next days, I think this could be one of the hot discussion topic or panel discussion in.
Yeah, it's a
hope
good idea. We'll convey it to the scientific team. But it's a
You
good
are the
topic.
scientific.
No, it's Alex and team. Yeah. Okay. Thank you very much.
Thank
And
you.
I'd just
And
like to inform everybody that on 30th at 8:30 p.m., we are having an online chat, literally, with none other than Dr. Palermo. He's agreed to have a very informal interaction. So looking forward to everybody joining in. We'll share the link soon.
How did you manage to get him in just a week's time?
I wrote to him.
Cool.
I emailed
He
him.
was so
He
reluctant
agreed.
to make, he was so busy with cooking stuff. Great to have him. Fine, cool.
We gave
Yeah.
him a new XC machine.
No, at first shot itself, he agreed. So, yeah.
Okay.
No, actually,
Thank
I
you,
told
Dr. Charu.
him
Thank
last
you.
year.
And Dr. Rajvi, Dr. Nishad, and everyone present here.